So just to be clear, this is the presentation that we have prepared among a group of these four faculty for the site visit. So I will be showing you other people's work. And this is really a high point of the history here. So I will take it at a slightly slower pace than I did with the site visitors, since we have the whole hour. I don't intend to take all of it. You may have questions. So let's talk a little bit about history. It is certainly true that this institute was really founded primarily for applying the methods of genetic epidemiology. So this is when Linda and Eves first came to DCU in 1980. I came in 1983. And much of our early work was actually launching a series of population-based twin studies that combined really for the first time some of the advances in modern psychiatric epidemiology, how to assess both disorders and environmental risk factors in large numbers of people. But then instead of limiting the kinds of analyses of twin studies that were typical at the time, which was like chi-square analysis, whether zygosity was whether concordance was greater in MZ versus DC twins, we instead began to apply the whole family of analyses that Linda and colleagues had developed in Birmingham. So we did begin molecular work in the mid-1980s, but I think it's fair to say that well into the mid to late 1990s, by far the bulk of the work that was done at this institute was in the area of genetic epidemiology. And we developed samples. So the Virginia Adult Twin Study of Psychiatric and Substance Use Disorders, the so-called ADD project, the adult follow-up, Chosen Twins, were a series of relatively large-scale studies, all of which were manned, run, interviewed, etc., here from VIPIG. But then soon thereafter, we began to develop a variety of international collaborations. And of course, many of you here have worked on these samples, so we've played sometimes substantial roles in the analysis of data from Sweden, from Norway, from Finland, from the Netherlands, from the UK, and from Australia. And we've often had trainees from the through our institute here, and ended up developing substantial collaborations. In addition to then developing the samples, there was a constant back and forth between the demands of the questions that the researchers wanted to ask and those who were interested in method development. So one feature of this program for a long time had been methodologists developing methods and want to try to apply it. Clinicians have questions that they want to answer that the methods are not currently there, and this is an ongoing dialogue over the years that has fueled a number of the developments that we have had here. And of course, this area is a continued focus of the training. So we as a group decided that in a roughly half an hour frame, there were 7 areas that we wanted to try to review, and again, presenting only the high points and the ones outlined here. So let's start with comorbidity. This was certainly a major focus of our work, particularly in the early years right after the adult twin study was completed. It was actually at that point difficult to write papers fast enough. Every single question that we asked, relationship between alcoholism and depression, phobias, each one of these were really new and novel questions at the time. An example of a particularly influential finding was evidence that actually was first found using Martin's Australian twin sample using symptoms, and then was found in several different forms of our adult twin study using lifetime diagnoses. Major depression and GAD had, to all intents and purposes, the same genetic foundation. About in 2005, there was a DSM-5 preparatory committee that was really founded on that work. It was all about the question about what should happen to GAD. So the rough metaphor there was that if mood disorders are mammals and anxiety disorders are fish, is GAD a porpoise? And that was the way the question was being framed at that point. I think the answer to some extent is probably yes. We also more recently, almost all of our early work at comorbidity, the unit of analysis was the disorder, mostly whatever the DSM edition was at the time. But the last 6 or 7 years, we've also done work at the criterion level, which I think is also quite interesting, but we will not be reviewing that here. So if we had to pick one of our papers that maybe best gives a broad overview, this is a collaborative effort that was done with Ted Reichhorn and his colleagues at the Norwegian Institute of Public Health. Again, we've all seen a variety of the Norwegian colleagues come through for the last 15 years nearly. So what Ted had done was to develop the first personally interviewed twin sample that had all the nice epidemiologic characteristics in the Norwegian studies. But in addition to doing the Axis 1 personality disorders, which we had done, although again not thoroughly, we did not do, I was always selective in the way we spent our time, that Ted really took a CD interview and basically applied pretty much the whole thing. But in addition, he also used an Axis 2 interview called the SID-P, developed by Bruce Fole at the University of Iowa, that assessed all 10 of the personality disorders that extant, which was a DSM-4. So we had really, for the first time in an informative twin sample, a representative set of the common Axis 1 disorders, not autism, not bipolar illness, not schizophrenia, which you cannot usefully assess if you only have about 2,500 people. You need many more to have informative numbers, and all of the 10 personality disorders. And so the question was, and I think I remember the discussion Mike and Steve and I had when we started this work, about how could we possibly analyze this. And I'm not going to go into the blood and guts of how this sausage was made. This was not using the standard and rigorous sequential kind of modeling that we're used to, and there is a bit more fuzz about that. I think that's appropriate, but this is not the time and place to discuss that in detail. This was the major set of results. This involved actually fitting a saturated Koleski model, then generated a matrix of genetic correlation that we then factor analyzed using, I think, GMN, oblique factor analysis. What was so interesting about this, the a priori question that one poses for analyzing data like this really can be formed to two extremes. One is the great level of non-specificity. There is, in fact, there was 50 years ago and now a resurgence of this idea that there is a G-like factor for psychopathology. That all psychopathology really represents is a single dimension of screw-uppedness, or whatever other name you want to use for that. And sort of got schizophrenia, autism at one end and some little mild personality sort of at the other end, but there's nothing qualitative about it. It's just a single dimension. The opposite extreme, which is probably even more unrealistic, is that DSM really absolutely got it right, as Moses was coming down from Mount Sinai, and every single one of these disorders represented a unique genetic disorder that had where the genetic variance was not shared. Having been in many of those committees, I thought the a priori probability of that was very low. So these results were actually conceptually pretty much in the middle of that design. What this is roughly saying is that you can explain that this is the genetic factors here. These four, that's a fairly pale. Use my backup here. Yeah, that's better. Four correlated factors. So notice that the interfactor correlations are pictured here, and none of them are extremely high, although the highest are between the two internalizing. And this broadly suggests that common psychopathology can be relatively well explained by an internalizing axis, right, that has the tendency to make ourselves suffer, divided between those that are by nature somewhat more episodic, that is, you get them and then you recover, versus those that are by nature pretty much chronic and are largely a part of the kind of person you are. And then externalizing disorders, which the general idea is you're making people around you suffer, which similarly are divided into those which are sort of episodic, but doesn't really apply to antisocial personality, and those that tend to be more kind of chronic in the kind of person you are. Now, I could spend a very long time talking about this slide. It's really quite rich and makes a lot of clinical sense. One of the most common concerns that were raised when this paper was submitted is how do we know that this is not entirely driven by method variance? We had two different interviews. They were done by the same interviewer. The patterning is a little bit different. The SID-P is sort of more broad questions about personality characteristics, but theoretically you can imagine that maybe the nature of the SID-P instrument just generated a series of different kinds of responses, and that was the major pattern of the factor. And I think the space evidence for that is not very supportive. And that's particularly true because the Axis 2 internalizing dimension includes 4 personality disorders, the ones in the black bound orders, that were assessed on the SID-P, but 2 of them are so-called Axis 1 disorders, but they're quite known for their chronicity. And in fact, many people think that social phobia and avoidant PD are sort of close cousins. They're not entirely the same. And dysthymia, as you may know well, but mostly dysthymic people are pretty much chronically unhappy. And similarly, then, the Axis 1 externalizing, you have antisocial PD fitting with these. One of the most interesting things for me was to see which were the disorders that didn't clearly fit. And one of them, just as you would predict, is borderline personality disorder, which actually sits midway between the Axis 2 externalizing. There's quite a lot of narcissistic and histrionic elements, but also those of us that have both clinically know, and if you do look at any of the genetic literature, there's actually quite a strong genetic relationship between borderline PD and antisocial personality that really shares both of those dimensions. And even more interesting is eating disorders. This is an epidemiologic sample, so this is almost entirely bulimia, anorexia being almost an order of magnitude rarer. in the general population, and anyone who's cared for bulimic patients, they're often quite dysphoric, rather like a classic internalizing, but they're also very impulsive. It's the act of the binge, and sort of this accumulated sense of internal dysphoria, and then this release, by going and getting 2 boxes of Kentucky Fried Chicken, or a dozen half donuts, and going to your apartment, closing the shades, and then stuffing yourself and vomiting afterwards. And that pattern is exactly replicated here by a substantial loading both on the internalizing and on the externalizing domains. So this was a broadly successful enterprise where we think we got, really for the first time, this sort of 30,000-foot view of what the broad patterns of common psychopathology are like. All right, so on to development. Development is a very good example within our institute where there's been an interrelationship between a range of structural models, particularly developed by Lindsay Eves and by Mike Neal, and then us applying data. And some of us, myself included, have stuck a lot with the very simple Koleski models, but a number of them have been considerably more fancy and more informative kinds of developmental models. Here, one of the examples I'll be talking about here is done by Dr. Edwards, and this was applying development, really an adult developmental question about the nature of the genetic vulnerability to alcohol consumption. And this was in the MM3 subsample of the VATSPOT study, where we had individuals retrospectively reporting using a daily diary study their average alcohol consumption per year from the age of alcohol initiation to the age they were at the current consumption. And here the rough clinical question was, is there just a single dimension that kind of turns on sometime in mid or late adolescence as alcohol consumption occurs, and that just manifests itself all the way going forward? A number of developmentalists, Tammy Moffat in particular, think that when you talk about externalizing disorders, you probably need to account for some set of more adolescent developmentally specific processes that occur. Think about the adolescent shit-kicking, or whatever sort of fancy framework you want to use for it, versus the, quote, rear or more life-term persistent kinds of developments that often start in the early 20s and develop over time. And so we were sort of wondering, would we see patterns like this within the sample of alcohol intake? And this is the simple Koleski modeling. We had good statistical evidence for 2 genetic factors. And those of you that are easy to do this, you see one of them has this monotonic increase over age, and the other goes up and then comes down. And if you picture that, I think this is a much more informative picture. You give these. So very much like Moffat would have suggested, think about what's governing genetically drinking in people like our Spit for Science project, right? The college students here. And really, it may be that you've got 2 processes. One of them is this kind of adolescent, very general externalizing factor. And underneath that is some potentially more malignant genetic predisposition that's much more likely to drive you, rather than being the adult experimenter who in your early 20s, you'll find a boyfriend or girlfriend, you'll kind of settle down, go to college, when you finish college, get a job, and stop heavy use and develop hopefully a more normative and successful alcohol use trajectory, versus those that are going to potentially have a greater probability of carrying on and developing more pathological alcohol use over time. So pretty simple model, but I would say quite clinically useful in helping us think about what might be occurring in alcohol use and alcohol use disorder. And that's really all I said. So another kind of set of developmental studies you've been doing are not actually strictly genetic epidemiology, they're more straight epidemiology. And this is something that Chuck Gardner and I have done, I guess, well back over 10 years ago. We seem to have this predilection for fitting these big developmental models we may get made fun of, and they think these are really either maps of the Los Angeles freeway system or of some kind of new transistor that we're trying to develop, but in fact they are classic structural models. And this was actually a continuation of the project, again, something that Dr. Edwards took the lead on, and this was from the ALSPAC cohort, this epidemiologic sample with born in 91 and 92, I think. And here our focus was not diagnostic, but it was alcohol problems at the age of 20. And we don't present many of the specific path estimates here, but it's only to note what was, and we have seen in the past, the prominence of an externalizing pathway to alcohol use disorder. So we actually boldly tried to model alcohol at 2 levels, at age 17 and 20. That's only 3 years apart. And note that that's a robust path. But the idea that you could really, that once you have alcohol problems at 17, none of the other risk factors are actually continuing to play a role is obviously quite robustly disproved. Because you've got a number of paths coming in conditional on age 17. A lot of these factors are still impacting on whether really at the latter stages of high school versus either as a young adult working or in college, that a number of factors are continuing to impact in addition to the alcohol problems at 17. And note the important role of the illicit substances. The highlighted ones here are roughly those that define this externalizing pathway, which has genetic influences, but really begin with this idea of conduct disorder in early adolescence or late childhood, and have these personality traits that are predisposing, that is the low conscientiousness, high levels of sensation seeking, again, high levels of conduct disorder now in middle adolescence that then predict substance use and illicit substances and other personality problems. So it's a good way of getting a sense about what the developmental cascade is that lead to these problems. All right, so now let's talk about familial transmission. This enduring question that we ask perpetually about the human condition, which is why do we resemble the people we grew up with? And over history, there have been a variety of different interpretations of that. I think it's actually only in the last 40 or 50 years that we've had the real infrastructure to answer those questions rigorously, but of course poets and playwrights have pondered this question from time immemorial. In fact, much of the work at this institute has focused on what we would call horizontal transmission because of the convenience and wonderful accessibility of twins, which are always of the same generation. But actually, I'm going to talk here more on a more challenging, but equally, if not more important question of vertical transmission. That how is it that parents transmit their vulnerability to psychopathology or substance use to their children or offspring? So there have been several designs that are able to do that. Remember, the standard design is entirely confounded. That is, you resemble your children, and there's no easy way within a nuclear family, despite many researchers' efforts to claim that it is, to determine whether resemblance between parents and offspring are a result of genetic or environmental factors. So one of the methods that we've used here, this is work of Dr. Judy Silberg, is the Children of Twins design. This has gone back another 25 years in a variety of different iterations. And here, this is the kind of path model that is being used. Those of you that are familiar with this, this is going to be, I think, moms, if I remember. Well, this is one parent, this is spouse, and these are the twins, which I guess either moms or dads. In this case, monozygotic twins, but it's the one for A. The space can be variable, and this is the second spouse. And so M is going to be the assortment parameter, and W is really the non-genetic transmission, while the 0.5 path is the classic transmission from A parents to A in offspring. So roughly, we're mostly interested in the question of the degree to which transmission is via environmental effects versus via genetic effects. And here we start with always looking at depression in the parents, and then look at two outcomes in the children. On the one hand, depression, so we're looking at prediction within a disorder, or predicting conduct disorder. And the rather striking results, which are delineated in this table, is that for depression, there in fact is no evidence of genetic transmission from parent to offspring. Now that's not the same thing as saying there are genes that are important in the parental generation and genes potentially important in the kid generation, but they ain't the same genes if they exist. And so rather radical in suggesting, at least in this sample, that now the resemblance is not that strong, flat off, but all of the resemblance that there within eras of estimation are environmental. And this is not an inconsistent finding. There are other parts of the literature that are beginning to show the importance of these kinds of parenting characteristics. And there's a variety of reasons why adult twin studies may be relevant and sensitive to those questions. But personally, the idea that there's, quote, no C for any major psychiatric disorders, although we often promulgate that, and it's certainly not true for externalizing disorders. I don't think it's true for depression either, but we haven't always been using those sensitive measures. Quite interestingly, it's a different model for conduct disorder. So here you have a sort of heterotypy in transmission, in which genes for depression in moms can predispose to conduct disorder with kids, and showing the potential complexity that we don't always expect that that genes are going to impact on the same phenotype across developmental periods. All right, so another far more classic method for looking at vertical transmission is adoption studies. I think I've mentioned to this group before that it's pleasing or ironic for me. My first ever genetic study was still when I was a psychiatric resident, and I asked Seymour Ketty if he would allow me to reanalyze the Danish adoption study of schizophrenia. So I spent many weekends driving up very early on Saturday morning from Yale, New Haven, where I was then living, to outside of Boston. I would have stacks of very thick personal interviews of the adoptees and their biological adopted relatives, and I was one, and I'd be scoring them. And here I am considerably later in my career through collaborations with a range of people in Sweden. Now being able to look at adoption studies, but actually in a considerably more powerful light, which we'll try to explain to you here. So the basic, using population-wide data in Sweden, there are approximately 18,000 adoptees with no biological relationship between the biological parents and the adoptive parents. Remember that it's pretty common. Mom and dad die in a car crash, aunt and uncle adopt them. So that quite a number of adoptions, in fact, are from our research purposes, really not very useful because of that confounding. And they have lots of biological relatives, and what you note is that this group of people now giving up adoptions, there's lots and lots of half-siblings. So both the biological mothers and biological fathers are rather, they took God's commandment to be fruitful and multiply, and they, with multiple spouses is very common. And adoptive parents are somewhat less. So in this case, we are looking at drug abuse, and here we are looking at, this is not an interview study, this is using administrative records. That is contact with a medical diagnosis, getting into legal trouble for drug abuse, not drug dealing, that doesn't count. Or pharmacy records, meaning that we know that you're doctor shopping and using multiple opiates or benzodiazepines. And we have lots of other data to suggest that these are at least reasonably valid, partly because they classify relatively accurately. So the major point, we do the standard, so there are two broad adoption designs. The most common one is the vertical design. I start with ill biological parents and see whether that transmits to their adopted away offspring, or I grab ill adoptees and I look in their biological and adopted relatives, and of course we can do both. And the first primary finding was that the risk of drug abuse in the adopted away offspring of biological parents was approximately double. When we then grabbed the ill adoptee, the drug abuse adoptee, we saw that in full siblings, and within confidence intervals, the same figure, approximately double the risk in biological siblings who they were not reared with. So again, having the same half-A squared kind of relationship. And in the half-siblings, it was 1.41, and of course, that's rather a nice ratio between the 2 to 1. But what immediately shows you how complex drug abuse is as a phenotype is that within sampling error, the risk in an adoptive sibling is not different from the risk in a biological sibling. So this is someone you were reared with, but not genetically related. This is someone you were genetically related with, but not reared with. So you both have approximately a doubling. Now, if you look more completely, and Hermine designed this slide, this is now looking at characteristics in the biological and adoptive families, all of which, this is the hazard ratio, showing the prediction of drug abuse in the adoptee. So let's start with the yellow, which represents the risk in the, actually no, let's start with the orange. That's the risk in the biological parents. So you see that drug abuse, that's the same 2 that I showed you before, but not only does drug abuse from the parent predict, but so does psychiatric disorders, so does alcoholism, so does criminal conviction, and actually even a little bit, so does biological mom's low education. Each of these are predictive of risk in their adopted away offspring. If we now look at the yellow curve, which is adoptive parents, that's not statistically significant because that's pretty rare, and because the confidence interval is large, but quantitatively it's not very different. But note that alcoholism in the adoptive parents significantly predicts, as does marital divorce, as does criminal convictions, and I think even this, death, is statistically significant enough that disruption of the adoptive parent bond predisposes. And then if you look in the siblings, this is now the adoptive sibling, and that's the biological sibling, and you'll also see in the biological sibling alcoholism also significantly predicts. So we've got evidence for the so-called genetic externalizing spectrum in this disorder. Genetically, drug abuse does not only transmit, but it gets transmitted with vulnerability to drug abuse and alcoholism, as we see in many of our twin and other studies. Now, part of the wonderful experience for me working with the Swedish colleagues is the opportunity to see whether we can get out of the box of the population-based twin study, which has been very useful, and how else can we find relationships in the general population that allow us to ask the questions we're interested in relatively novel ways. And there's a fairly long lineage of this project, but in essence the insight first was that there are quite a number of people in the general population who are functionally like the biological parents of adoptees, which I mean that they inseminate the kid, or much more rarely in mom, they donate the egg and are pregnant and deliver, and afterwards have no other relationship with the child. men do that 10 to 1 compared to women. We call them deadbeat dads, we call them not living fathers, define them in a variety of ways. And there's quite a substantial number of people who function like adoptive parents. Those are step-parents. Nothing especially fancy about this. So what we were able to do is to define families where they had a mom, and this is most easily described here. So mom is the linchpin of these so-called triparental families. She gives birth to the kid, she raises the kid, and she's with the kid for at least, actually for all of the first 15 years. And this is now the not live with father who inseminates mom and then never lives with her or nearby in the same small community. And then the kind of hero, if you want to think about it, is the stepdad who comes into the family and lives with the family for at least 10 of the first 15 years. So to a rough approximation, remember this, what's unique about this, there's no adoption registry, there's none of the atypicality, the adoption screening, this is occurring naturally, there's no legal process here, this just happens in human populations that these families occur. But to a first approximation, they represent rearing environment plus genes only, and rearing environment. And so the simple result here is we looked at these for all three of the externalizing disorders that we've been looking at in the Swedish population, and we see the same ordering between them. These, again, are hazard ratios. So that mom's drug abuse most strongly predicts drug abuse in the kid. Next, the not live with dad predicts, but less than mom. And next, the stepfather. And notice that this ordering is very similar across all three. So it suggests to a first approximation that both genes and environment are important. But note that we can estimate the environment in two ways. This is a direct measure of it, and the difference between those two is another measure. And we actually have done this in path modeling, which many would argue, and probably correctly, is a more appropriate way to do it. And to a rough rule of thumb, these are additive. Otherwise, if you look at the tetrachord correlations, this plus that come pretty close to summing to that. So there's no complex, massive interactions between myriad and genetic effects. Okay. Genotype environment interaction. So first approach is actually to look at the same study. And here we took an aggregate multiple regression model of genetic risk from everything we knew about the biological relatives and looked at that related to an aggregate risk of the family, the adoptive family, from the adoptive relatives. And then we looked at, fit them as linear models, and then we looked at an interaction on an additive function. So this is an interaction on the additive scale. So we're looking at risk differences, not risk ratios. And this was the result we showed, which in my words is kind of a classic fan-shaped distribution. So this is from the most benign adoptive environment to the most predisposing, and these are individuals across deciles of genetic risk. And the key finding is that those at least genetic risk, these guys here, basically they change absolutely less than 1% in their risk when going from the most benign to the worst. By the way, the worst adoptive environment in Sweden is a pretty damn good environment. It's an absolute term, so it's not like we're talking about sort of horrific family experiences. While those at high genetic risk increase over 4 percentage points. So roughly, these guys are gaining about 4 times as much in absolute terms in risk, under the assumption that here the genetic variants are both predisposing to an average risk, but making these individuals more sensitive to the pathogenic effects of the environment. This kind of classic fan-shaped interaction. Now, another very different approach, and this is one that we as an institute, along with many other places in the world, have been moving into, is beginning to incorporate into our genetic epidemiology designs molecular measures of liability, of which we know there's GCTA, there's polygene risks, and this was done by Dr. Jessica Salvatore, in which we were asking the question, going back to the ALSPAC sample we talked about, could we revisit prior work that both Dr. Dick and I have done on genotype-environment interactions for alcohol-related problems, where we have shown a range of factors, particularly parental monitoring and peer deviance, that high levels of parental monitoring seem to be successful at suppressing the effect of genetic risk. Right? You've got to really, well, I think that, I won't tell the psychological story, but I think it's fairly intuitively clear. While peer deviance acts in the other way. If you have a propensity toward alcohol abuse and you are surrounded by, all your friends like to go to prayer service and talk about Sartre, you're less likely to manifest disorders. While if everyone else thinks that grabbing a bunch of stones and going on the highway on Saturday night and throwing them at cars going by is really a great night's entertainment, you're much more likely to manifest your genetic risk in the latter social environment than you are in the former. And we wanted to try to test that now, instead of using the standard latent variable modeling from relatives using polygenes. And lo and behold, this is, by the way, a proxy for parental monitoring. So, do you know where your kid is at 9 o'clock on Saturday night? And that's kind of the standard question. And lo and behold, to statistically significant levels, those at high polygene scores, there's a greater difference with low parental monitoring, while high parental monitoring really suppresses the difference, just as we found more traditional measures. And here again, the same thing as in reverse, as you predict. High pure deviance allows the genes here indexed by polygenes to have a greater effect. So this is, I think, a quite pretty example of the kinds of things that we like to do. All right, so structural modeling. So this is really tries to show how sometimes theoretical developments drive data collection, and sometimes data collection opportunities drive theoretical models, and that's kind of a dialectic that has gone back and forth over the years, and this one is more of the latter. So as we discovered, Dr. Moss has worked with us closely in our Swedish collaborations, that we realized that we had some kind of new opportunities, some of which were just more relative. So this is now a list of the population-based sample of MZ, DZ, full SIB, maternal half-SIB, paternal half-SIB, first cousins, and step siblings that we were able to analyze for this particular sample. And our programs are not used to having 7.6 million pairs and how they fit in. So there was just a practical problem here about utilization of such large samples, and we can try to correct individually for ascertainment, all that other sort of stuff, which we ended up not doing. I'm not going to try to defend that here. This is the same drug abuse definitions that we utilized previously in the adoption sample. But we realized that there were other things we could do, interestingly, mostly to teach us more about C. C has been a pretty poor step cousin to A in our twin models. Like most of us are sort of happy if it's not there. We sort of say, we're really geneticists, we want to find A, we can figure out ways of twiggling and making the C kind of go away. That's often fine with us. I think for externalizing disorders, that's just not the case. Because these are disorders where environmental transmission is actually quite an important phenomenon. And we realized that we had really two pretty good natural metrics in the Swedish population that allowed us to determine further what C might be doing. This is the kind of modeling that Dr. Moss developed, in which we moderate the C and potentially A covariance by age differences. Now, there's a long theoretical discussion that we need to have now that there are certainly some circumstances under which you may want to model A by age differences, and I think a good rationale, which we can go into if you want. Here we're using it as a measure of C. Let me just be very brief. The main point is that A moderation really makes sense if everybody's getting an assessment at the same time. So I can really say, oh, this person is 12 and they're getting assessed, and this person is 16. So between 12 and 16, there might be developmental changes. In the Swedish data, everybody has years and years and years to get a disorder. So, so the idea that there's going to be any systematic difference. Now, theoretically, you could dig in and using ascertainment, maybe there's some small differences in years, but that general model, in my view, doesn't apply so well. There's that maybe debatable. But in this case, we modeled it as a function of age differences, and then, because we have data on this, we actually know the length of cohabitation. How many years did they actually reside in the same family together? And so the goal was really to parse out C, to see whether we could figure out more what underlies that for drug abuse disorder. So these are the major results. For example, we really are able to very confidently discriminate that we really have both qualitative effects from genes and qualitative effects from the shared environment. And to a first approximation, the shared environment is a little more different for drug abuse between boys and girls than the genetic effects. These are the overall estimates here. We do see there being a little bit more shared environment for males, and we've seen that in several other data for drug abuse, although not all for alcoholism for reasons that are obscure to me. But the most interesting are these curves here. So what we have down this axis is the years of age difference. So the dark blue, of course, are twins born in the same year. These guys are sometimes called Irish twins, right? Lamont quickly reproduces right after the first kid. And you can see that there's slightly less, and this is a function of the years of cohabitation. So this would make sense particularly for siblings. And you see that cohabitation here is actually a fairly robust effect. And this is consistent with other analyses, in which the C-squared is really substantially modified by the number of years you're living in the same household. So one very easy way to see that is the difference between the relationship in the Swedish population for which we have most power to look at cohabitation effects is half-sibs. Because the vast majority of full sibs live together, and almost all the twins live together. But in fact, there's large groups of half-sibs, which is somewhat confounded in that paternal half-sibs are
less likely to be reared together than the maternal. So theoretically, you might have interuterine effects, which the maternal have some share and the paternal don't. But but we have quite a bit of power, and very few cousins are raised together. So, but notice that we're really able to disentangle these effects and show that they are both of significance, as both the number of years reared together and the age difference does partially index the C that is relevant for difference. Okay, now meta-analysis. So we've actually had a long tradition of doing meta-analyses. Mike and Pat Sullivan and I think in the early 2000s, published a couple of meta-analyses, both on major depression and schizophrenia, and every once in a while, as I think most of you do, I look at how my articles are doing over time, and those are kind of well up into the sort of 800 or 900 citations. I mean, these have been very, very commonly reviewed, and still represent the definitive analysis, and I'm glad to see that we haven't shot our water entirely on that, but Mike, Brad, and I recently published one on alcohol use disorders. This, in fact, is even a level up in sophistication, because we did the lazy thing of going through old papers and trying to abstract into a classic meta-analysis, which in some cases made you crazy, because the earlier twin studies don't always provide necessary information. Hermine did us one lot better, and actually through United Funding did a mega-analysis, in which actually got to go with investigators, collect all the data. It is way more work, figure out how the questionnaires are sort together. And here I'm only going to take a minute reviewing that. The phenotype here is the sort of most basic one, which is smoking initiation. Here's all the samples. And this is what the aggregate prevalence looked like over time. So all you see is this kind of monotonic increase across all the various colored samples, that on average, the number of people who initiate cigarettes increases through adolescence. A stunning finding. And you can see there are obviously some population differences between them, but there's a very clear aggregate line. More interesting was fitting standard ACE models and their variation by sex and age. So I'm going to, for the sake of argument, assume that this age 12, which is very, very wide confidence intervals, is really not something we know very exactly. And we'll point out that excluding that time point, we see this lovely monotonic trend for heritability to increase over adolescence, and that has been seen in many other samples and makes all kinds of intuitive sense. But equally interesting, given how important C is, we find a parallel declination in the importance of C. So these two are really mirror images of one another. That in the early stages of adolescence, when you're initiating, cultural and shared environmental factors are quite a bit more potent on your decision to smoke, and as presumably more smoke gets into your body, your genes do the sorts of things they do, that the actual decision whether to use becomes much more a result of genetic factors. Now E is not doing very much, but interestingly, this is the C correlation between the two. And what you see, although it takes a touch of imagination, is it looks like there's a bit of a spline. It's pretty flat and very high, up to around 15, then it drops. Now, and do boys and girls start doing something more different from each other at the age of 16? Well, yeah, they may be. Like girls going out with older boys, and of course, give them cigarettes and other nastier substances for certain benefits. So there's a set of potential influences that might be occurring here that are going to be different between the sexes. All right, so the final category is correlative designs, and this has been a very active area. I would fair to say has been a source of preoccupation of mine for a number of years, partly because of the wonderful opportunities afforded by my Swedish collaborators. But it can be used in a variety of ways to disentangle this foundational issue about correlation versus causation. And here we're going to be talking about studies that Dr. Silberg did in the VATSPUD study, looking at this important and increasingly interesting area of bullying. So let's just start with the standard epidemiologic data. These are risk factors at an epidemiologic level in this cohort of bullying, being bullied, not being a bullier, although that does also have its effects, and the major psychiatric disorders. And we see it's actually not uniform, so it doesn't predispose to everything, but you can see those with the asterisk, so the significant effects, and it's particularly suicidal ideation is really, it's tragically highly correlated. Major depression, ADHD, separation anxiety disorder, not seasonal affective disorder, those are really confused, and social phobia. No, is that social phobia? So, or social anxiety. Okay, social anxiety. So those were the ones were just a flat up relationship. Now the problem here is the obvious one. I was going to say young adolescent boy on the playground. I kind of noticed what bullies do. Bullies are not random in the respective people they pick on. By and large, they pick on those who are more vulnerable, both physically smaller, not very assertive. They rather have an easy triumph, not get themselves bruised. So when you see psychopathology correlated with bullying, you really cannot, despite the tension to do that, leap to the causal interpretation, oh, those poor kids, they got bullied, we need to go out and prevent bullying. It might work, but if in fact the bullies are very specially selecting people, it's not going to work. The bullying is really maybe more epiphenomenal if there are other personal dysfunctions. Now, we were never going to do a controlled trial of bullying, so we should never have to think that we're going to get a definitive measure of this. And whether social psychologists might have tried to get that past the IRB, I think that would probably be problematic. So one of the best things we're ever going to find, not perfect, is the co-relative design. And here, this is the kind of patterns that Judy found, where we see 3 out of the 4 of them are, although notice how very wide the confidence intervals are actually statistically significant among co-twins. And these were MZs, or both MZs and DZs?
MZs.
MZs, all right. So this is really complete matching for genes and complete matching for rearing environments. So while this does improve causality, it certainly gives a large uptick of our confidence that bullying may actually be a causal influence on these predispositions rather than just an effect of the bully being very careful and wanting to select anxious kids who aren't going to fight back overall. So a very interesting pattern of results. Now this last set of slides are future directions. I think that's actually less relevant for this group, so I'm not going to go through these. And I did take a considerably more leisurely pace, given that I have a whole hour to myself. So there is quite an opportunity for questions if you have any. I mean, the people to thank for this are the people I listed on the first hand. There's lots of others involved, including Swedish collaborators and others, and Norwegian folks.
Ken, is the future of twin studies bright? And how do we need to change our game to maintain our competitiveness?
I remain a firm believer that the techniques in our quiver of using statistical and population-based methods are complementary to those of molecular genetics. They serve somewhat different purposes. If you asked me and I were to review another grant whose major goal was for $2 million to estimate the heritability of major depression from a twin study, I would think that that's not a very good realization. But what I think you have seen over time is that we have, through increasing conceptual and statistical sophistication, sometimes matched and sometimes demanding better data, been able to ask increasingly informative, complex questions from this data that have yielded influences that are important and influence the field. I give talks about these in one way, so we see how journals have treated them. So I don't think there's this sense, that twin studies are dead. It's Mark Twain who said that it's often over-advocated. So yes, do we have to think hard and cleverly about how to ask more sophisticated, more relevant questions? We do. Should we be in a position of sometimes trying to add molecular genetic variants to our twin studies and be able to utilize, as we're now developing, to look at our standard latent variable models with the ability to index genetic vulnerability for molecular markers? Absolutely. And should a broad research program try to include both methods? Absolutely. That is what we're trying to do. We have to judge how successful. Okay, well, I think that means you all get to go out 15 minutes early. I'll hang around for a few minutes if you have any questions. Thanks for your time.